This is chapter 5, part 5. Some of this chapter I pulled from the PowerPoint, so don't get too excited if you don't see stuff in the PowerPoint that might be mentioned in the book. So, I wouldn't stress over that information. If it's not in this PowerPoint and it is not in my screencast, then don't worry too much about that. Doesn't mean some random thing might not come up on the test, but I try to avoid stuff that I don't focus on in the screencasts. All right, so techniques in cultivating and identifying animal viruses. Viruses require living cells as their medium. So in vivo is lab-bred animals and embryonic bird tissues, and in vitro is cell or tissue culture methods. Primary purpose of viral cultivation, basically to research. I don't even have to get into this. It's to figure out what is making people sick and what we can do to prevent it. That's basically why we cultivate viruses. At least the official reason why people do it. So tissue and cell culture techniques, again, I took out some bird and animal stuff out of this, and I just focused it on this because this is the main useful information. So isolated animal cells are grown in vitro in cell or tissue cultures rather than in an animal or egg. The cell culture or tissue culture is grown in a sterile chamber. Specific media is in there that contains the correct nutrients for the cells to survive. There's a monolayer or single confluent sheet of cells that forms, and this allows for the close inspection of culture for signs of infection. In order to detect viral growth in a culture, there's an observation of degeneration and lysis of the infected cells. So there's plaques, they refer to them as plaques, areas where viral infected cells have been destroyed. So that's a visible manifestation of the CPEs. So this is an example there. This is a cell culture and a cell culture, or these are cells before the viral infection, and then this is after, and you can see that there's destruction there. Prions are primarily protein. This is kind of another variation of a virus. Exact mode of infection is still being investigated, but it does deposit long protein fibrils on the brain of tissues of humans and animals. Examples are the Creutzfeldt-Jakob disease, afflicts the central nervous system and causes a degeneration and death. Bovine spongiform encephalopathy, mad cow disease, Shy-Drager syndrome, or multiple system atrophy resembles Parkinson's. So they are linking these prions to these diseases. Viroids are virus-like agents that parasitize plants, about 1/10 of the size of the average virus, composed of naked strands of RNA lacking a capsid or any other type of coating. Significant pathogens in economically imported plants, tomatoes, potatoes, cucumbers, citrus trees, and chrysanthemums. Viruses and human health. So common causes of acute infections: colds, hepatitis, chickenpox, influenza, herpes, warts. These are virus-caused acute infections. Worldwide, dengue fever, Rift Valley fever, yellow fever, all caused by viruses. Infections from viruses with high mortality rates: rabies, AIDS, and Ebola. Infections that cause long-term disability: polio, neonatal rubella. Connections to chronic infections. They're linking viral infections or chronic viral infections to type 1 diabetes, MS, various cancers, Alzheimer's, and obesity. So again, way better to prevent these viral infections than to suffer from them and end up with something else later. So antibiotics are designed to treat bacterial infections and have no effect on viruses. So if you've ever had a youngster that had an ear infection, chances are the doctor said, go home and let it, play its course, let it have its course, because 9 times out of 10, it's or a sinus infection or something. It's something that just needs to run its course. It is very difficult to find drugs that will affect viruses without damaging the host cells at the same time. Almost all antiviral drugs licensed so far have been designed to target one of the steps in the viral life cycle. So if it interrupts that life cycle, then the virus is unable to replicate. It is definitely easier to develop vaccines than to prevent viral diseases, and that is an absolute fact. So last part, antibiotics are an effective method of treating viral infections, true or false? I gave you the answer there. I hope you all figured that out by now. So that is the end of chapter 5. Thank you for listening, and if you have questions, let me know.